First of all, I'd like to thank our chairperson, Dr. Huber, and I'm from Japan, ninja world from Japan. I'm not a ninja, but a basic scientist. So we're no.
I was in Japan.
Oh, really? Okay. So we are now doing their basic science, basic study about their innate immune response against cancer and infection. Today, I'm going to talk about a short talk about our recent data regarding application of innate immune activation to cancer therapy. As you know, it is known that pattern recognition receptor mediated recognition of our pathogen-associated molecular patterns triggers activation of our innate immune response, such as induction of innate cytokines, which also leads to subsequent activation of adaptive immunity for efficient eradication of pathogens. And recent great series in the studies of innate immunity has revealed that not only toll-like receptor, but also quite a lot of other pattern recognition receptors, such as Reguai-like receptors, are involved in this system. And during the virus infection, virus derived nucleic acids are mainly targeted by certain pattern recognition receptors, that is, nucleic acid sensors. In the most cases, such a viral sensors activates type I interferons production pathways for antiviral activity. In particular, Reguai, retinoic acid-induced gene I, and MDA5, melanoma differentiation associated gene 5, are key cytoplasmic pattern recognition receptors for the detection of RNA viruses, including influenza virus, hepatitis C virus, measles virus, and so on. And the virus derived 5' phosphate RNA and double-stranded RNA are ligands for Reguai and MDA5 respectively. So ligand binding activates their downstream signaling pathways to induce type I interferons and pro-inflammatory cytokines for triggering antiviral innate response. On the other hand, activation of the innate immune system are actively pursued for the therapeutic application in oncology from two major points of view. As you know, innate immune activation gives rise to both indirect and direct anti-tumor effects. As for indirect anti-tumor effects, for example, pattern recognition receptor ligands can be used as a vaccine adjuvant or potent immune modulators. As for direct anti-tumor effects, certain pattern recognition receptor ligands show direct tumoricidal activity on tumor cells. So in this study, we focused on the direct anti-tumor effects mediated by Reguai and MDA5. Because it is known that Reguai-like receptors such as Reguai and MDA5s are ubiquitously expressed in non-immune cells as well as immune cells. So we first stimulate the various kinds of various types of cancer cells with the triphosphate RNA and poly-A-C, both of which are the Reguai-like receptor ligands, and then we examine their interferon beta response. Interestingly, MCF7 and A5/9 cells showed a much stronger response as compared with the other cancer cell lines that we tested. So we focus, in this study, we focus on the breast cancer cell. The next, we further examined several other types of, several other breast cancer cell lines, like MCF7 cells, T47D and ZR75-1 cells showed robust interferon response, both messenger levels and the protein levels. On the other hand, in contrast, certainly interferon beta response was not observed in MDA-MB231 cells. So we hypothesized that the type 1 interferon receptor mediated signalings was impaired in these breast cancer cell lines. But however, MDA-MB231 cells show rather higher levels of OAS messenger RNA, which is all known as one of the interferon-inducible genes in response to type 1 interferon alpha treatment. However, they fail to induce both interferon beta and alpha beta messenger RNAs. And next, we found that these three breast cancer cell lines, which showed a robust interferon response, appear to be morphologically dead. On the other hand, such an effect was not observed in MDA-MB231 cells 48 hours after treatment with these regarding receptor ligands. And we also quantitatively evaluate these results by triple staining. So around 50% of each of these three breast cancer cell line cells were dead. But MDA, MB231, and MRC5 cells did not show any damage. And we also confirmed that confirmed by the annexin 5 staining that apoptosis was actually induced in regard receptor ligands treated MCF7 cells. And back to the previous result, these three breast cancer cell lines can induce a robust interferon response. On the other hand, MDA5-MB, MDA-MB231 cells cannot. So we next hypothesized that there may be some correlation between interferon response and the cell death. And we next examined the effect of the type of interferon and on like receptor ligand-induced breast cancer cell death. As you can see, treatment with anti-type 1 interferon receptor blocking antibody suppressor like receptor ligand induce cell death in MCF7 cells. So these results suggest that like receptor ligand induce MCF7 cells death is largely dependent on type 1 interferon receptor mediated signaling. So in the next setting of experiment, we also examine whether the interferon treatment itself can induce cell death or not. But unexpectedly, treatment we could not observe significant cell deaths in response to a single treatment with a type I interferon beta, even a high dose of our interferon beta. So finally, we are also examining the effect of regular receptor ligands on the tumor growth in nude mice. As you can see, our MCF7 cell inoculated mice showed tumor suppression in response to these treatment with these regular ligands, but not interferon beta alone. On the other hand, MB231 cells in inoculated mice hardly showed such a tumor suppression in response to any treatment. So this is a summary. So based on these data, we speculated a model as shown here. So our current data suggests that the type I interferon induces cancer cell death requires regard-like receptor-mediated signaling. So without regard-like receptor signaling, interferon do not induce cell death, but at least in MCF7 cells. So in this respect, our data may partly explain the reason why the single treatment with type I interferons has limited efficacy for most types of cancer. So in relation to this, I'd like to introduce one interesting previous old paper, which described the type I interferon-mediated antiviral activities. So their results suggest that type I interferons can induce apoptosis in virally infected cells, but not in non-infected cells. In this case, interferons makes the cells resistant to viruses. So at that time, 1998, any innate sensors were not identified yet, but now we can understand that virally infected cells give rise to pattern pumps induced signaling, but not but the non-infected cells don't. So these results are somewhat consistent with our results. So that's why I showed that our ongoing project, we're now working on the detailed mechanism for regular-like receptor-mediated licensing to the induction of cell death in response to type I interferons. And we also studied about whether our current data are also treat all other types of cancers, and whether there is a difference in regular-like receptor ligands induced cell death between cancer cells and non-cancer cells. And I'd like to reassess anti-tumor activity induced by type I interferons and find some key to enable efficient optimal cancer cell death by interferon treatment. The last slide shows all the collaborators to this work. I'd like to thank all of them. Thank you very much for your attention. No, still. So many researchers are studying about this point, but well, everyone says everything, anything, yes, in at various points. So yes, yeah. Sorry. What's the relation between interferon and innate immunity? Sorry? What's the correlation between
interferon and innate immunity.
Interferon and innate immunity. Colism. Well, in general, activation of a pattern recognition receptor induce a type I interference in downstream, and which induce the cell antiviral state. And the last paper suggests that their interferon can induce apoptosis only in infected cells, but not to non-infected cells. Yes.
Now there is correlation between the genetic expression and the interferon expression.
Yeah, right.
So interferon can induce apoptosis by two ways: by enhancement of granzyme A and granzyme B, this one pathway, and at the same time can increase the expression of trail and trail binding receptor.
Yeah.
Also, trail can induce interferon expression. So
Yeah.
Direct correlation between both of them.
Yeah. I think so. Yes. Thank you very much. But our paper add some point to that. Yes. So there is some licensing mechanism. So we're now working on that. Yes. Yes, please.
So innate immunity, it also includes some non-pathogenic autoantibodies. Did you ever notice any connection between the presence of autoantibodies and cancer?
Autoantibody and cancer. In terms of innate immunity. Well, that's a very maybe for example, type I interferon is very important for the B cell maturation. So maybe some excessive activation of our innate immunity induce excessive type I interferon, and which may contribute to our autoimmune production or antibodies production. Yeah.
You did look at those?
No.
Are there other questions for Dr. Takaoka?
Thank you very much.